Mainly his research interests are in bioluminescence, biochemistry, luciferase reactions, and luciferin biosynthesis pathways, hormonal regulation and physiology of bioluminescence, evolution of bioluminescent traits. He has published seminal articles in very, very highly acclaimed and impactful journals, including that journals in Science Advances and Scientific Report and other, to name it. He is the recipient of the Prix Ars Electronica Linz honorary mention with Nero Akamatsu Award in the year 2023. We welcome Professor Yuichi Oba. Please, the stage is yours, Professor Oba. Will be grateful if you can confine within 40 minutes of your talk. Thank you.
It is a great honor for me to be invited to this excellent conference. So, my name is Yuichi Oba. I'm from Japan. My expertise is bioluminescence. Maybe you're not so familiar. The bioluminescence is the light produced by the living organisms. Actually, the specialists of the bioluminescence are not so many in the world. And to my knowledge, there are only a few biologists focusing on the bioluminescence in India. So I understand this conference is the interdisciplinary. So today I'd like to introduce our recent activity and how fascinating the bioluminescence science is, especially for the young Indian students. First, the bioluminescence research starts by discovering new bioluminescent organisms from both terrestrial and marine fields. Japan is a small country, but fortunately, its moist land is surrounded by the deep ocean, and we can easily access various terrestrial and marine organisms. Recently, we discovered the bioluminescence of the springtail in Tokyo and Yokohama. The body size of this creature is about 1 millimeter. When stimulated, many spots on the dorsal side glow in green. The luminescence of a springtail was actually recognized as early as the 18th century in Japan. But it was completely forgotten. We realized that this species is already known species recorded in early 20th century from Yokohama, Japan. Thus, we concluded that this species, Lobella zauteri, is bioluminescent. The many Japanese newspapers covered this topic. We also surveyed new bioluminescent organisms from the world. Recently, we discovered new bioluminescent land snail was in the wood of Thailand. Previously, only one species of bioluminescent land snail was recorded in the world from Singapore, though our survey in collaboration with the taxonomic team in Thailand, we discovered additional species of bioluminescent land snails. This topic was covered by many Japanese newspapers, and unexpectedly, this snail won the prize of the International Mollusk of the Year, 2024. As an additional prize, the whole genome of this land snail was sequenced by the International Society of Mollusk Research. Have you ever seen the luminous spider? Why am I asking you? Please take a look at this. This is an old publication in Nature, 1925. Dr. Burnham Brown a curator at the American Museum of the National History, reported discovering a luminous spider in Myanmar. However, he couldn't capture this specimen, and this record was almost forgotten. But please take a look at this post. Very recently, it was posted on the social media that a luminous spider was snapped in India. He also failed to capture it. That's why I was asking if you have ever seen a luminous spider. If we can capture it, it will be a good news. And yesterday, I learned from a student of the Central University of Kerala that he told me the Kerala is famous for the bioluminescent beaches. The source of this luminescence is not bacteria, it's a tiny plankton called Noctiluca scintillans. And I also learned yesterday that this becomes famous for the movie Kambarangi Night. Yes? And also find the article. I'm not sure the growing mushroom is toxic or not, but I think it is not Philaboretus manipularis, because this species is also distributed in Japan and looks completely different. So I think this species in India might be the member of the genus Omphalotus. Japanese Omphalotus species is highly toxic. Next, we are also studying on the reaction mechanism of the bioluminescence in various species. The bioluminescence mechanism of the luminous mushroom has long been undetermined. However, from 2015 to 2018, in collaboration with Russian group, we revealed the true bioluminescence mechanism of the luminous mushroom. The bioluminescence in mushroom is continuous. And we found that enzymatic cycle continuously rotates in the mushroom using these 4 enzymes. This is the mushroom luciferin. The mushroom luciferin is oxidized by the catalytic activity of the luciferase to produce the green light. And this oxidized product was converted to the caffeic acid, then to the cisplatin, and again to the luciferin. And it is interesting that the gene of 4 enzymes are tandemly aligned on the same chromosome. Now, please pay attention to this compound, caffeic acid. Caffeic acid is an essential compound in plant because it is a precursor of the biosynthesis of lignin. Lignin is one of the primary components of the cell walls of the plants. So all plants possess caffeic acid. Because of this fact, we can expect that if these 4 genes are introduced into the plant, it will start growing like a mushroom. And this is indeed true. The growing petunia is already on sale in the United States. As shown in this slide, the elucidation of the bioluminescence mechanism is sometimes valuable for creating new tools or products. Now we are trying to elucidate the bioluminescence mechanisms of luminous land snail and luminous springtails. That was we discovered. In the land snail, we still cannot determine the luciferin molecule and the luciferase enzyme gene. However, some reaction mechanisms have been determined. The enzyme is insoluble, but can be extracted using a detergent. The reaction requires hydrogen peroxide. The bioluminescence mechanism of the springtail was also not determined. However, we found that reaction requires magnesium and ATP, like the case of fireflies. In future, this bioluminescent mechanism will be completely elucidated, and they may be used for creating some tools and products. Next, we are also studying on the physiological regulation mechanism of bioluminescence. This is a fish we discovered very recently. This is unpublished data, so I'm sorry I cannot say the species name here, but this is very interesting. Most bioluminescent fish inhabit in the deep sea, but this species lives in the shallow water. The luminescence reaction mechanism of this fish is still completely unknown. At least this bioluminescence is not bacterial. However, we found that luminescence of this fish can be induced by the treatment of melatonin. Why melatonin? So our next interest is the biological function of bioluminescence. What is bioluminescence function of this fish? This is my hypothesis. This fish lives in the shallow water and has a white belly. The white color functions as a counter shading during daytime, like in many other shallow water organisms, such as tuna and penguins. They hide their shadow with their white bellies. At that period, the melatonin level is low. But on a moonlight, the weak moonlight creates the faint shadow. At that night period, melatonin level is high, and this fish grows weakly on its belly to hide its faint shadow. This is a strategy called counter-illumination. Thus, we think the induction of bioluminescence of this fish by melatonin can be explained for the counter-illumination at moonlight. Please imagine this fish growing weakly in the shallow water at the moonlight. Do you agree it is difficult to see? Meanwhile, the bioluminescent function of the luminous land snail is difficult to imagine. For mating, like firefly, maybe not. This land snail grows strongly in juvenile than in adults. For attracting prey like the anglerfish, no, this snail is herbivorous. And this is my hypothesis of the bioluminescent function of the luminous land snail shown in a cartoon. The firefly larvae eat land snail. At that time, the larvae generally grow. And the luminous land snail might pretend to be snail eaten by firefly larvae. And probably firefly larvae hesitate to attack the luminescent land snail because it looks like the snail that is being eaten by firefly larvae. And the land snail will survive. And that is my hypothesis. Now I'm trying to reveal the true function of the bioluminescence in the land snail. Experimentally, is a group in Thailand. As shown in the previous slides, we are studying various bioluminescent organisms. However, the most well-recognized bioluminescent organisms is, of course, the firefly. These are Japanese firefly, Nippon Rukiora cricciata, flashing in yellow-green on the river. This place, Toyota Town, is famous for watching firefly in Japan, not for automobile. It's a different city. Many people visit there every year to watch this bioluminescent show of firefly. Firefly have been beloved in Japan from old days to now. This is an old Japanese wood block print over 150 years ago. During that period, people from the wealthy to the ordinary poor already enjoyed watching fireflies while eating and drinking outside at early summer night. And the children's, also young ladies. The title of this print is Looks So Happy because she caught flying firefly by hand and is feeling happy. The culture of deeply appreciating firefly is probably unique to Japan. But 100 years ago, we collected huge numbers of the firefly for sale. And in 1970, the water pollution destroyed many firefly habitats. And rapidly, the number of the fireflies decreasing. So now the firefly is one of the most important icon or symbol of the conservation and species protection in Japan. So the research on the firefly is fundamental topics for the conservation biology in Japan. This is a very interesting survey about what Japanese people think. This survey, there are several questions on insects. This question, what is your most favorite insect? Now, the best one is firefly. The second best was the rhinoceros beetle. And the third was stag beetle. Maybe you don't understand why. But anyway, this is also interesting. Which insect would you feel lonely without? The best one is firefly again. And the second and third were changed to dragonfly and bell cricket. Now you will understand how Japanese people love fireflies. Now, it is considered that over 2,000 species are present in the family of Lampyridae, firefly, and it is interesting that the color of the luminescence depends on the species. For example, this European common firefly, Lampyris noctiluca, emits in green to yellow-green. And the North American common firefly, the Photinus pyrolis, emit in yellow or the orangish yellow. And another North American firefly, Pilactonemena angulata, emits in the deep orange. Now the question is, which color did the common ancestor of firefly emit? And we challenged this question. Molecular phylogenetic analysis revealed that the common ancestor of firefly appeared about 100 million years ago. There are more direct evidences. This is the first firefly fossil in Burmese amber from 100 million years ago, reported in 2015. There is a light organ-like transparent window here at the tip of the body. Probably this is the light organ. And in 2024, the second example of the firefly fossil in the Burmese amber, dating about 100 million years ago at the Cretaceous period, was reported. This species exhibited a more distinct light organ here. And the later part of my talk, I would like to introduce this topic, resurrecting the ancient grove of firefly, published in 2020. We revived or resurrected the light of the ancient firefly that appeared 100 million years ago in the test tube. What did we know it? First, I would like to share the basic knowledge about the firefly bioluminescence. This is the luciferin of the firefly. luciferin, this compound is catalyzed by the luciferase enzyme in the presence of ATP and magnesium and oxygen to produce the oxidized luciferin and light. And the color of the light varies in fireflies, and the luciferin is common in all firefly species. And the color of the luminescence is determined by the amino acid sequence of the enzyme luciferase. This is the sequence, the amino acid alignment of the American common firefly and the European common firefly. They emit yellow and green respectively. So you can see that most of the amino acid is conserved, but some amino acids are different. In other words, these differences in the luminescence color, the differences in luminescence color is caused by the microenvironment of the luciferin binding in luciferase protein. In this study, we revived ancient luciferase using ancestral sequence reconstruction strategy. This is a brief explanation of how to perform the ancestral sequence reconstruction. First, amino acid sequence of the genes, specifically luciferase in this study, are collected and performed multiple sequence alignment. Second, the phylogenetic tree is reconstructed to determine the ancestral protein sequence. In this study, we used the maximum likelihood algorithm under the JTT substitution model. Of course, ancestral sequence reconstruction is not our new method. There have been several precedent researches using ancestral sequence reconstruction. For example, in this paper, they revived the ancestral rhodopsin, a light-sensitive receptor of the dinosaur age, using ancestral sequence reconstruction. They revealed that the dinosaur eye were most sensitive to the green light in the dark. It is actually not a surprising result because the most of the extant vertebrates also have the most sensitive eye for the green light. And go back to the luciferase. The ancient luciferase gene calculated by ancestral sequence reconstruction strategy is synthesized, and the recombinant protein is expressed in this study in bacteria E. coli. And as I mentioned, luciferin molecule is common in all fireflies, so it is undoubtful that the ancestral firefly also used the same luciferin molecule. And the luciferin is mixed with the recombinant ancestral luciferase, and the luminescence intensity and the spectrum are measured. Now I'd like to show our ancestral sequence reconstruction result about ancestral luciferase. We collected over 30 gene sequence of firefly luciferases and their homologous genes. Then we made multiple sequence alignment and performed the maximum likelihood phylogenetic analysis. We revived 7 ancestral sequence alignment, ancestral luciferase in our paper, those the blue circle. But to simplify the story, we focus here on the luciferase of the common ancestor of the firefly here, unclamp. This is the inferred amino acid sequence of unclamp. This graph represents the probability distribution of the amino acid residues. The probability of the most residues in unclamp were almost 1. And the average was about 0.95. It means that our ancestral sequence reconstruction is reliable enough. Then the recombinant protein is expressed in E. coli and mixed with the luciferin. And this represents the luminescence activity. The luminescence activity of the unclamp is comparable to the extant luciferaces. And this represents the spectrum of the 7 ancestral luciferaces. This will be difficult to imagine the color, so the luminescence color was photographed. And this is the result. As shown here, the Ankh lamp emits a deep green color. And this is the movie of the luminescence of Ankh lamp in the test tube for demonstration. And this is a comparison between the Japanese extant firefly luciferase and Ankh lamp. Maybe you will recognize the color differences. So now we concluded that the first firefly in the Cretaceous period emitted in green. Please remind, the dinosaur eyes were most sensitive to green color. Maybe the dinosaurs watched fireflies. From here, we consider the protein structure. In 2006, a Japanese scientist, Dr. Nakatsu, showed the X-ray structure of the Nipponorukiora cruciata luciferase with the luciferin analog, DLSA. They also showed the X-ray structure of the cyto-directed mutant of Nipponorukiora luciferase with serine 286, asparagine, which emit in red. In the luciferase protein, isoleucine 288 interact with the benzothiazole ring of the luciferin. And in wild-type luciferase, this benzothiazole ring is tightly sandwiched in the hydrophobic pocket by isoleucine 288 for minimizing the energy loss. On the other hand, mutant luciferase, there is more space between luciferin analog and isoleucine, and they discussed that less rigid microenvironment allows some energy loss from the excited state, probably due to the thermal relaxation or the organization of the specific interaction, and lead to the emission of low energy red light. Now about ancestral luciferase, which we made. We also performed X-ray structural analysis of the Ankh lamp with luciferin analog DLSA. This is the superposition of Nipponorukiora luciferase and Ankh lamp. The aromatic ring of the phenylalanine interact to the thiazolene ring of the luciferin in the wild type. In the wild type, the valine 243 pushes the phenylalanine from behind. And the distance between thiazole ring of the luciferin and phenylalanine side chain, benzene ring, is 3.43 angstrom. But in the ancestral luciferase, valine is substituted to the more bulky amino acid isoleucine. And isoleucine pushes phenylalanine more tightly to luciferin. And the distance between the luciferin and the phenylalanine the side chain is 3.18 angstrom. So we think the more rigid microenvironment enable further minimizing the energy loss to give the green light in Ankh lamp. We published this work in 2020, and I titled this paper as Resurrecting the Ancient Glow of the Firefly. The word resurrection has a special meaning for European people. It means the revival of the Jesus from the grave. And this paper was covered extensively in newspapers and science magazine and TV. However, no newspaper questioned why green. In this regard, I have an idea, and I wrote in the discussion section of the paper, like as the general terrestrial predators have eyes most sensitive to green light. And the original biological function of the luminescence of firefly is considered to be the warning display. As a consequence, we can say that green light emission is most advantageous for ancient fireflies. Look at this. These are photos of the typical terrestrial bioluminescent organisms. They all emit in green. The probably true enemy of the ancient firefly was our ancestor, nocturnal insectivorous mammals like shrew, not dinosaurs. Indeed, various mammals, including human, feel bad taste for firefly, and they never eat firefly. Probably those old memory luminescent insect is not edible. It's still inherited in mammals, and firefly are survived till now. Today, I introduced my recent activities and how fascinating bioluminescence research is. This country, India, is very rich in biodiversity, and I think there are many undiscovered bioluminescent organisms in here. Also, as I introduced today, the bioluminescence research contains many scientific topics that are still not fully understood. I hope many researchers take an interest in this field. Let's work together. Thank you for your attention.
Thank you for a very illuminating talk, in fact. So let's pass over to the audience.
Hello, doctor. Thank you for nice and interacting talk. Thank you for nice and interacting talk.
Thank you very much.
Okay. So, in the phylogenetic tree reconstruction, why did you not take Ant-Elad as the most recent common ancestor? Ant-Elad. There was a common ancestor of all Ant-Elad. Why you did not take it for account?
Ant-Lac.
Sir, you can show phylogenetic tree.
Anklam. This is
Phylogenetic tree.
phylogenetic. Sure.
Yes, this one. Ant-Elad there. Ant-Elad. Yeah, yes. Why you did not take this one? It is the common ancestor of all.
Yeah, this is the most ancestral sequence of the superfamily Ratalidae. So this ancestor is probably non-luminescent, and yeah, no light.
Okay, so I was just wondering when you could figure out the molecules, how easy or difficult it is to synthesize by the chemist in the lab.
You mean the luciferin?
I mean, these are the natural ones, right? So whether it can be synthesized artificially in the chemistry lab.
You mean the synthesizer of the...
These fluorescent compounds, whatever the new natural ones you are getting, whether it can be synthesized by the chemist in the lab.
Synthesized in the lab or not?
You mean the...
Can we synthesize these compounds in laboratory?
Yes, sir.
Synthetic.
Yeah, synthetic gene.
So
And
it can
luciferin is also synthesized.
Chemically synthesized.
Chemically synthesized. Luciferin is chemically synthesized.
So anyone ever tried? For the natural, no, the number have been tried, but like whatever you have shown, any one of them have been tried, because then the direct translational prospects can be there to use them as a labeling dyes.
Professor Rava, what about yellow fluorescence? Yellow fluorescence, Because some of them emit yellow.
Which one?
Yellow color.
Yellow.
Oh,
Yellow.
This one?
some flies emit yellow fluorescence.
Yeah, some species
So
are.
where they situated in the evolution? It's after green.
Why? That's important. For the firefly, which emit in adult, the light color is important for mating. So if the partner can recognize the color, any color is okay. So probably the first firefly emitted in green, but they switch to the purpose for the mating, and they start changing the color. And the luminescence, the firefly which emit yellow, the partner can detect the yellow most sensitive. And the firefly which emit orange, the partner can detect the orange color most sensitive.
And
that's the reason.
Just one more also. You said that mushroom, you recycle the the firefly and to
mushroom.
caffeine in mushroom. But the luciferin the firefly is not recycled. Why? It's because the living capacity. Because this mushroom recycle, but the firefly do not recycle.
Yeah,
Maybe due to living or due to chemical nature.
Yeah, usually the bioluminescent creature have a lot of luciferin, but in the case of the mushroom, it contains only small amount of luciferin. They produce the luciferin gradually for continuous growing.
Yeah, wonderful talk, sir. So I was wondering, so you have spoken about the loose structure of the luciferase and a tight structure of a luciferase, which creates the difference in the color. So I was wondering, do you see any species which has multicolors or different colors because of the gradual change in the structure of the luciferase in any organism that is naturally present?
You mean the toxicity of the firefly?
No, any species which is having different colors, like the same organism having different colors of emitting.
Very rare. In the case of the firefly, actually the firefly pupa, they emit two different colors. In the pupa, they emit yellow in the tail and faintly green in the body. So that's the only rare case. But usually, one bioluminescent creature can produce one color.
Stage?
Yeah, sometimes depend on the life.
Sometimes. So, thank you again for Dr. Yuichi Oba, professor in the environmental biology. Now it's.
Thank you, sir, for the informative session. Now I request Dr. Rajendra Pillangata to present the memento as a token of our gratitude.